So we moved over here to this other management near Jason's farm, and I went ahead and filled out this left side where it says soil map units. Since this is the same soil type, I looked at it on Web Soil Survey before we came. So it's an Orangeburg, just like the one we just left. So I filled that out, and then soil moisture would be moist here because the weather conditions are the same, correct?
Same. Yep.
And so with that, I guess the first one we need to look at is soil cover.
Soil cover.
And it's you know, whether we got any residue, cover greater than 75% estimated, and by the looks of here.
So in looking at this one in terms of surface cover, this one is quite a bit different than the one we looked at in that adjacent field in the different management. This is a small grain that's planted, and we can see that there is some existing residue, but we do see quite a bit of bare soil throughout that's not in rows.
It's not 75% though.
Not 75% cover, so you're thinking that we are less cover?
Less. It says surface cover from plants, residue, or mulch. Okay. Cover greater than 75%.
Right. Yeah, there's a lot of bare ground. One thing that could maybe help us, even though this is an estimate, one thing that could help us, we could do that Transect method to count at each individual point. It would really get us in closer to a more specific number. But again, with it being an estimate, you know, it looks, just looking across this field, like there's a tremendous amount of residue. Right. But when you really get down in between the rows and weighing all that out, it actually may be 50% or less.
Yeah, that's a good point. When you look at, you don't want to look across the field, you want to look straight down to check residue.
Mm-hmm. So in terms of the criteria for this one, I would say that we probably are less than 75% of surface cover.
So no, it does not meet.
Does not meet. We can actually move on to the next indicator and look in terms of residue breakdown. Again, back to what we're looking for in terms of that criteria is natural decomposition of the crop residues or organic mulch is expected with cropping conditions. So we have small grain here. It looks like it may have followed corn of last year.
I also see some, here's a peanut. There's some, I've seen a few vines. Okay. So there's still some, and I imagine they plowed this field, so the old residue probably has got buried.
Mm-hmm. Yeah, see a corn cob here. You're finding peanuts there.
That's so two seasons.
Two different seasons. We do have corn stubble that, again, we have stalks that are buried halfway in and halfway out of the soil, so definitely looks like there was a tillage pass. Even though they are there, we do see that the stalks are broke up, whether it's been done by the biology or if it's actually been done by the tillage operation.
You can actually see where it was cut, where the probably turbo tail or vertical till tool cut
that spacing looks
residue.
Looks like it coincides to that spacing of those coulters. So we can see, of course, with a peanut being a legume, the seed-in ratio is going to be quite a bit lower. So you wouldn't expect that much of the vines be around.
Right.
But you did find a hull, so that's
a few little pieces of vine, but not much.
Okay. So you would think that a lot more of that residue would be gone. And digging into our little soil pit here at this point, I can break this apart. It breaks apart really easy. So again, that kind of leans more towards that this operation had some type of tillage prior to
prior to the small grain.
Small grain?
After the corn.
So they may have used that tillage to
prep the ground for the drill.
Prep the ground, condition that residue, and then planted the small grain. But I don't see very much, if any, residues that have been buried at this point. Of course, we could dig several other places. We see residue that is submerged into the soil profile in multiple places. But in terms of breakdown, you know, the thing is natural decomposition of the crop residues or the organic mulch. So, you know, we see something
a lot of these corn stalks look like they were just put here.
Just put here.
Very little decomposition.
So we see more effects of condition of the residue was more tillage implemented than maybe the natural decomposition.
Correct.
So I would think that this criteria would not be met for this indicator. Okay, so we can move on to surface crust, and there's this is a pretty good indicator, and we can actually, unlike the other management, we actually see quite a bit of visible surface of the soil. And when we think about that crust, you know, that soil being exposed to the elements, then those surface crusts can actually form. And anytime that we dig here in the soil surface, we can see that a surface crust has formed either from an irrigation vent or rainfall or other weather events. So we can see in terms of our criteria for surface crust, crusting on no more than 5% estimated of this entire field. And we can see that anywhere that we dig where the soil is exposed, that we have a crust. And we think back to the surface cover or the soil cover, you know, we had a 50% is what we were thinking, so the 50% of the soil is visible, so we're definitely easily over 5% of this entire field
is crusted.
That has surface crust. So we would also be no in terms of meeting that criteria. Ponding or infiltration, that indicator, no ponding or on non-hydric soils within 24 hours. We say that had that same rainfall event, this field is relatively flat. We don't see any evidence of ponding, but if we look around,
it's pretty sandy soil too.
Yeah, sandy soil. So this would be an opportunity that we could do the infiltration ring to see if it meets those criteria. Or looking around any little depressions, we could see if there are materials that have bridged up. But we would assume if we have surface crust that during a rainfall event, it is likely that we could see some ponding for some period of time, just because we understand what surface crust will do in terms of holding water out of that soil. But in terms of
since
ponding,
we don't see it here today, though, would we just mark no?
I would say, or we would say yes, that it meets the criteria because we don't have evidence of it being here.
Gotcha.
You want to do the penetrometer?
Yeah.
And see what we get here? And then I can also use a wire flag to see if we get similar results. So in taking this, we're just going to see in multiple places if we find penetration resistance.
150.
150.
Huh, it's pretty close.
Pretty close to the same depth that we're finding.
So that's 4, 5 inches.
Mm-hmm. Got a little bit deeper there.
A little bit. That was about the same.
So you're finding a little bit shallower compacted layer than I'm finding with the flag.
But I think it's still gonna meet because you're not making it to a foot, huh?
Right.
So, or not meet, I should say.
Not meet. So penetration resistance, penetrometer is less than 150
within the top 6 inches
and less than 300 in the 6 to 18.
So that does not meet the criteria.
Correct.
Yeah. Now water stable aggregates.
Water stable aggregates. So we have our pit here, our sample hole.
Oh, we don't have the water, do we? Oh, yeah, we do.
We have our water. We're going to do the same test that we did across in the other management at that evaluation. So we'll take surface. And we'll crumble it up into, oh, there's a little bit of buried residues. And place it in the strainer. We'll submerge that in to our water. Allow that to fully saturate. A few air bubbles coming out. Okay. Bring that up. Okay, again, in comparison to this one, but we'll look at water stable, aggregates, and the strainer. Soil structure remains intact with aggregates apparent.
I guess if you're going to run this assessment, it's a good idea to do it on several different areas, because if I hadn't have seen what the other one did, I wouldn't have known exactly how bad maybe that one is.
Mm-hmm. Because
so once you run them a few times, you kind of get the feel for.
Yep. Because this one, in comparison, has slumped.
Right.
It's sunk down quite a bit, and it was very evident that we saw aggregates in that sample because it stood up and the water left that sample. We do see some water leaving this sample, but everything looks to be kind of just hazed over, or real shiny, like we're not seeing any aggregates at all. And it has squatted down considerable. So in terms of meeting this criteria, I would say no as well. But what we could do to compare this, we could actually take some of these clods, if we're able to get a clod, and let them air dry, and we could check those in the tubes as well to see if it would withstand sitting in those tubes. And
it's like the slake test, kind of?
Yeah, the slake test. But we have to have air dried samples for that test. In regard to, we can use field soil at field conditions with this strainer method. So moving on, in terms of soil structure, again, this is still mapped that Orangeburg, loamy sand.
Loamy sand.
So this, again, as I mentioned earlier, that usually with sandy soils, we're not really focusing on soil structure, just because of the nature of sandy soils. But we can see that this one, it does fall apart really easily. But it's more just very sandy. When we look at it pretty close, we don't see those obvious aggregates that we would be looking for, and that kind of reinforces the strainer test that we did there.
It's gone down a good bit since.
It's still slumping down. So in terms of soil structure, we may not use this indicator.
Right.
And that's the beauty of this evaluation, is we don't have to use every indicator in every evaluation, because this soil it's really sandy, so it's not going to really support identifying that structure. In terms of soil color, we did those comparisons to.
Very much so was a difference.
Yep, the field edge in comparison with this management of the Orangeburg and the other management of Orangeburg, and we did see and noted multiple differences. So looking at the criteria, no color difference between assessment area and fence row. That's not the case. We saw huge differences.
The answer's no on that
in those comparisons. Biological diversity, of course, we can dig around.
Look at plant roots too while you're digging.
And look at plant roots while we're digging. So we can pop us out a sample and we can dig through here. Looking around, there's not much residues to rake back, but we could identify to see if we see any soil life. In this case, if we had a hand lens, we could utilize a hand lens to hold this up a little bit closer to see if we could identify anything really small. I'm just thinking back into the other plot that we did. There was quite a bit of insects that were flying around while we were in the other evaluation. We're not seeing those flying insects.
Only thing you might find here is a fire ant.
A fire ant. And hopefully I won't be sitting in.
You won't find those.
So plant roots, they don't appear to be restricted in any means. but I'm not seeing very much soil clinging to those roots in that clod. And in our digging around in here, we do see a little bit of soil clinging to some of these roots. We would hope to see some. But we're not finding any earthworms or other soil life while digging through our actual sample area.
So possibly for plant roots, we might could give that a yes, because
I would think so. Plant roots. We do have some of these that we are pulling up that we do see a soil film.
And we do have at least we have a living root here.
We do have living roots, right. In the row. Yeah, we have some soil clinging onto multiple roots there, closer to the base. Maybe not these finer roots on the fringes, but definitely on the roots inside.
So we didn't find the three different soil type. Three different types of organisms though.
That's right. We haven't found any in terms of trying to assess the biological diversity. We look closely. We don't see anything crawling around. We're not finding any earthworms in digging through.
No pores, biopores either.
Also, that would be evidence of not being able to find those biopores, correct? Because we're not finding them. And you have to think about, well, why are we not finding those? With this soil being very, very loose, we could understand that there was that tillage path. So if there were biopores present,
they would have got crushed.
They would have got crushed in that tillage operation. That also could have impacted quite a bit of that biological diversity as well, especially the larger.
Earthworm doesn't do well with a disc.
Exactly. They're not going to be able to take that type of that type of disturbance. So in terms of biological diversity, we're not seeing three different types or more than three different types. So I would say that we do not meet that criteria. And
biopores neither.
Right. On biopores, I think we also do not meet the criteria for that one as well, just based off of they were probably destroyed within the tillage operation prior to this wheat being planted. So that gives us all of our answers to a majority of the indicators, all except for soil structure for this evaluation point. And now we can take that information and use it to work through the decision tree for identifying any resource concerns that we have at this evaluation point.